Good morning, everyone. Today I'll be talking a little bit about N-glycosylation in the brain. And in the first part of the talk, we'll talk about N-glycosylation in the brain in general, so looking at the importance of N-glycans in neurobiology, as well as some of the features of brain N-glycosylation that make it unique, and then finally looking at some of the factors that can shape the brain N-glycome. In the second part of the talk, we'll focus on the human brain N-glycome. So what are the conserved features and what are the derived features that make it unique? And then finally, we'll just touch on some of the factors that have shaped the evolution of the brain N-glycome in mammals. So let's firstly look at carbohydrates in the brain. So actually, they make up only around 1% of the total brain mass. And most of those are attached to lipids in the form of glycolipids. And N-glycans, which is the subtype of glycosylation I'll speak about today, you can see they make up only a fraction of 1% of total brain matter. But that doesn't mean that they're not important. I'm sure I don't need to convince anyone in this room about the importance of studying N-glycans, but maybe not everyone is aware of all the important functions that they play in brain physiology. And they're critical in the normal functioning of all neural cell types. Here are just some of the critical processes they are involved in. So it should not come as a surprise then that altered N-glycosylation is also associated with numerous brain disorders, some of which are listed here, but this list is growing all the time as people become more interested in understanding N-glycans in brain physiology and disease. You can see it's a rapidly growing field, but as is the case in many fields, large-scale large-scale N-glycomic studies are still lagging behind the other modalities, as you can see. And it's only in the last couple of years that large-scale N-glycomic studies of the human brain have really taken off. So it's early days, but we have learned some things about brain N-glycosylation so far, and one of them is that it's quite peculiar. The brain has a very characteristic N-glycoprofile, largely due to some very unusual structures which are not present or rarely present in other tissues, and structurally they're very unique. So that presents a challenge when we're trying to structurally annotate the brain N-glycome, because not only do you have to think about all the regular types of glycans, we always have to consider these tricky ones as well. The other thing we know is that the N-glycome of the brain is not static. In fact, it's dynamic and malleable, which seems logical enough to glycobiologists, but actually when we talk about the brain, it's kind of a newer finding, because the prevalent view up till now was that it was quite static and stable. But we now know that there are many factors and many stimuli that can shape the brain N-glycome, both intrinsic and extrinsic factors. Some of them are shown here, and they influence it in various ways, positively or negatively. We won't go into that today, but we're learning more about it all the time as more studies are being done. And this knowledge that the brain N-glycome is malleable and responsive is kind of like a double-edged sword. Because on the one hand, in the context of pathology, it can be a bad thing, because we know that there are certain triggers or certain stimuli which can negatively impact the brain N-glycome and lead to pathology and impaired brain function. So that's the bad side. But the dynamic nature of it provides hope that if we learn how to harness some of these properties through the use of targeted glycan-based interventions, we might be able to one day manipulate the brain N-glycome for good to try to shift this back towards a healthier type of brain N-glycome and potentially restore brain function. So that's really the ultimate goal, and that's why we study N-glycosylation in the brain. And that brings us to where we are today and why we have a special interest in the human brain N-glycome. And of course, comparative neurobiology is an approach to help us understand the human brain and what makes it special. Of course, it's an age-old question. This kind of approach where we study the human brain side by side with our closest ancestors has brought us many interesting insights from other fields of molecular biology about the human brain. But what about N-glycomics? Are there maybe some human-specific features of brain N-glycosylation that might explain our unique traits? Well, let's review the published literature, and it won't take very long because there's really been only one, I would say, meaningful comparative brain N-glycomics study published so far, which was this one I mentioned earlier. And it was a very important study because it was the first of its kind, but the conclusions were a little bit underwhelming because they basically conclude that the brain N-glycome is highly conserved, which it is. And they compared the mouse and the human brain. And that's absolutely true, but we felt that there's more to this story and that some things in the study could maybe have been done differently to flesh out some of these details. So we've tried to address some of those things in our study. So firstly, while they compared only 2 species, we've expanded this now to 4, including 2 non-human primate species, so macaques and chimpanzees, to get a better handle on which evolutionary changes are human-specific. Because when you compare just mice and humans, you don't really know when something's different, whether it's human-specific. It could well be mouse-specific. Secondly, whereas they only compared 1 brain region, we've expanded this to 4, covering all the different anatomical subdivisions of the brain. So we have the cerebellum from the hindbrain, the striatum from the basal ganglia, the hippocampus from the allocortex, and the dorsolateral prefrontal cortex from the neocortex. Because our goal was not only to identify human-specific specializations in N-glycosylation, but to also anatomically pinpoint where they are happening and where they occurred, because that could give us a better idea about their importance and potentially also their function. Regarding structural resolution of the glycans, while they reported only monosaccharide compositions, and we know how ambiguous that can be, we've tried to go into a lot more detail about structurally defining the glycans that we discovered, including doing linkage-specific derivatization of sialic acid so we can get information about interesting things happening with sialic acid linkage. Finally, we integrated our glycomics data set with complementary transcriptomics, both at the bulk tissue level and also single nucleus RNA-seq level, to look at the interaction between glycogene expression and the brain N-glycome phenotype. So let's start with the glycomics, let's start with the transcriptomics data first. And what we discovered was that actually glycogenes, or I should say their expression, in the primate brain is extremely well conserved. We looked at a subset of genes, 46 glycogenes, who are predominantly involved in N-glycan synthesis. And when you compare the conservation of their expression to 46 randomly selected genes from the genome, the glycogenes shown here with this red line are always at the extreme end, meaning they're very, very well conserved in primates. And what's interesting is if you break this down to the single cell level and look at individual cell types, most of the divergence, which is shown here in yellow, it actually happened in the non-neural cell types, so things like blood cells, endothelial cells, effectively contaminating tissue when we're talking about brain tissue samples. If we look at the actual brain cells, the neurons and glia, their expression was much more highly conserved. So they're really, really very well conserved. And this is what the correlation looks like of glycogen expression between the 3 primate species. So they're very well conserved. And if we compare that to the correlation of the N-glycome globally, you can see the correlation of the N-glycome is lower for each of the primate pairs than it is for glycogen expression. And then if we look at the correlation of glycogen expression and the brain N-glycome phenotype, that interaction is even less well conserved. So altogether, really, this data is telling us that the primate brain-end glycome evolves faster than the underlying transcriptional framework. And that's not something that revolutionary. It's something that glycobiologists have been saying for a long, long time, which is that N-glycosylation is a medium for rapidly generating additional interspecies diversity. But it's nice that we've shown that this is also the case in the brain. So now moving on to the glycomics data, and what did we discover by analyzing all these brain regions in all these species? Well, firstly, we found a lot of different glycans, nearly 300 distinct N-glycan structures across all species and regions. But interestingly, only 60 of those were ubiquitous, in other words, present in every region and every species. And so we call this the brain N-glycan core network. This is what it looks like when you draw it out. And just looking at it quickly, you can see that they're mainly simple, small N-glycans, mainly neutral ones, very few silylated ones. Some of them are complex, but a lot of them are non-complex, so oligomannose, porcimannose, hybrid, and also truncated, which is a class kind of specific to the brain. And they're all more or less biosynthetically related through one enzymatic step. And so we think this is the dominant N-glycosylation pathway in the brain, and that's confirmed as well by their abundance, which takes up 75% of the total brain N-glycome. So they're extremely overrepresented. So these 60 glycans take up 75% of the abundance of the whole brain N-glycome. And we hypothesize that they're expressed by ubiquitous brain proteins involved in brain homeostasis and keeping things in the brain ticking over. And that leads us to this model where the other 25% of the brain N-glycome is much more diverse, many more structures here, and we suspect that they're involved in regional species-specific functions. Of course, we need to investigate that further. But this is what a typical brain N-glycan profile looks like. On UPLC, we're showing the human PFC, but all the N-glycomes looked very similar. In fact, when we look at the composition of all of the 16 brain N-glycomes, they are quite similar, as you can see straight away by eye. So yes, the brain N-glycome is very well conserved. It is highly conserved. But if you dig deeper and you scratch beneath the surface, there are a plethora of interesting both anatomical and phylogenetic differences. So a lot of variation, which we'll touch on now in the next couple of slides. And there are two main trends that I'll be talking about, and they involve, firstly, a gradient in N-glycome complexity. So here we're talking about the abundance of complex N-glycans. And secondly, we will look at sialic acid linkage, so a switch from alpha-2,3 to alpha-2,6 linkage. On this slide, we are showing evolutionary changes in brain N-glycosylation according to different taxa. So firstly here we're looking at features of the primate brain N-glycome, so shared with the primates but different to rats. Then we're also looking at features specific to the hominids, and the last column shows us human-specific features of N-glycosylation, in other words, different to the other primates. And for each of these, we're showing them for all the brain regions. So there's a lot of information on this slide, too much to unpack now, so we'll just focus on a few things. But so these are the statistically significant differences that we found. But interestingly, there are a lot of things here, but you can group them into some similar trends, okay? And the two main trends were the ones I mentioned before. A switch from non-complex to complex N-glycans, and the other one is related to a switch from alpha-2,3 to alpha-2,6 link sialylation. And you can see that in the human lineage, most of the differences are related to this switch in linkage. So let's look at that first of all. If we just look at the proportion of complex N-glycans that are sialylated, throughout evolution there hasn't really been much of a difference in this proportion. But if we focus in on just the sialylated N-glycans and take a look at their linkage distribution, we see that there's been a gradual shift going from rats to hominids, where there's been increasingly greater usage of the alpha-2/6 linked variant. And we have evidence for this if we move back to our transcriptomics data, where this is single nucleus RNA-seq data. And when we looked for human-specific patterns of glycogen expression, we found enrichment for expression of alpha-2/6 sialotransferases. You can see the red dots show enrichment in the human lineage, and 3 of the genes with the biggest red dots were actually alpha-2/6 sialotransferases. They're expressed in different neuronal cell types, and of course we need to investigate that further to confirm that that actually results in an increase in these kinds of glycans. But there's evidence that this switch in linkage is encoded in the transcriptome in humans. The final evolutionary trend I'm going to talk about is this gradient in complexity. So here we're looking at the PFC, the dorsolateral prefrontal cortex, and we can see that as we move from rats to hominids, there's been a gradual linear increase in the abundance of complex N glycans. And this was true not just for the PFC, but in all of the regions we investigated. So it seems to be a global trend, a brain-wide trend in increased N glycan complexity. And when we investigated the rate of change for this, it seems that it's been a gradual process. None of the changes deviated too much from the expected rate given the evolutionary distance between the organisms. So there haven't been, in other words, many jumps in any species. It's been a gradual process throughout evolution. So that brings me to the end of my talk. Just to quickly summarize, what have we learned about the brain and glycome? Well, in general, the mammalian brain and glycome is very well conserved. We've even identified now a core network of N glycans. And having said that, even though it is well conserved, it does evolve more rapidly than the expression of the underlying glycogenes. We found human-specific cell-type distribution of glycogen expression, and we can say that we've identified a couple of evolutionary trends in the human lineage. So we can say that human brain evolution has been characterized by 2 main trends: a global increase in N-glycome complexity, as well as a shift towards increased usage of alpha-2/6 linked sialic acid. Of course, we don't really understand yet the significance of these trends. It's something that will no doubt form the basis of future investigations, but that's the direction we'll be heading in the future. And just to finish up, I'd like to thank all the people involved in the study, and I'll end here. I think we maybe have 1 or 2 minutes for questions if anyone has any.